Hello, we are a subgroup of Bioinformatics are going to present to you about a look on the demonstration of application of bioinformatic resources of blast protein. Moving on, we're going to look at a brief introduction of blast protein. So what is blast protein? BLAST is actually an acronym for Basic Local Alignment Search Tool and refers to a set of programs used to generate alignments between a nucleotide or protein sequence, which is referred to as query, and nucleotide or protein sequences within a database, which is referred to as subject sequences. On the other hand, BLAST protein finds regions of similarity between protein sequences. The program compares protein sequences to a sequence databases and calculates the statistical significance. Most of the time, BLAST protein function as to infer functional and evolutionary relationships between sequences, as well as to help identify a protein.
Now we are going to show a demonstration of how BLAST protein can be used in molecular biology or biotechnology in terms of identifying a protein sequence. First, go to the NCBI webpage, and then choose the data and software from the list menu on the left side of the home page. And then choose Tools in the Data Software side and click on the Basic Local Alignment Search tool. Then click on the Protein BLAST. This will be the protein sequence that we are going to identify. Then paste those sequences in the query box. And then choose the standard database and BLAST P protein BLAST before clicking the BLAST button. This will be the result displayed on the screen, and then we need to analyze and look at the percent identities which describe how similar the query to the aligned sequence. Hence, the highest percent identities will be chosen for protein sequence identification.
Next, we will demonstrate on using BLAST to compare two sets of protein sequences using pairwise alignment method. Firstly, use the link given to go to the NCBI webpage. Under the popular resources at the right side, click BLAST. Proceed to click at Global Align. In the Global Alignment webpage, do not forget to choose protein as we are going to compare two protein sequences, not nucleotide. These two examples of sequences will be used to do the comparison. The first protein sequence should be pasted into the box for query sequence, while the second protein sequence should be pasted into the box for subject sequence. Do make sure that the sequences are in continuous form and there are no spaces between them. After that, click BLAST. You can then look at the description box below to identify the second protein sequence. Then, click on the alignments and change the alignment view to pairwise with plots for identities. This will allow us to view the comparisons between the sequences easily. To compare the two protein sequences, observe the gaps, mismatches, and also the percent identity. That will be all for the BLAST mechanism.
Next, we will be focusing on the evaluation of usefulness of BLAST protein when compared to other similar softwares. The software chosen is FASTA. FASTA can be used to find protein sequence similarities where it is good for finding related sequences. The similarities of both databases are both allow protein sequence comparisons with protein databases, and both use pairwise comparisons for sequence alignments. The differences between the databases are in terms of global or local alignment. FASTA uses local sequence alignment first, then extends to global alignment. Meanwhile, BLAST-P uses local alignment. In terms of type of search, BLAST-P thrives in searching highly similar protein sequences, while FASTA excels at searching less similar sequences. In terms of sensitivity and speed, BLAST-P categorizes proteins approximately 6 times faster than FASTA, since it searches for only the most significant patterns. And for the significance part, bioinformatics is increasingly using FASTA, but BLAST-P or BLAST in general is still the most prominent similarity search method.
Embossed needle. The similarities of this software with protein BLAST is that both are pairwise sequence alignment tool for protein analysis. For differences, embossed needle use global alignment while BLAST-P use local alignment. In terms of sensitivity and speed, BLAST-P gives faster results as it uses heuristic approximation. In type of search, embossed use USA and UFO while BLAST-P use accession number or FASTA format. That's all from us. Thank you.